Hi, I'm Dr. Ed Pistilli, and I'm in the Division of Exercise Physiology at West Virginia University. In today's lecture, we're going to answer the question, how does skeletal muscle contract to produce force? To begin this discussion, we first need to understand the structure-function relationship of skeletal muscle. And what this means is the structure or anatomy of skeletal muscle is specifically related to its function, which is force production. In the image here, the two athletes on the left are producing high force power contractions. In contrast, the two athletes on the right are performing low force contractions, but have the ability to repeat these contractions over a long period of time. It is the structure-function relationship of skeletal muscle which allows these athletes to be successful in their respective sports. To begin, I want to break down skeletal muscle into all its functional parts, and I want to introduce that terminology now. At the outermost level, we'll start with the whole muscle, and the first level we can break that down into is the muscle fiber. As we're going to see, the muscle fiber is the cell of the skeletal muscle tissue. The muscle fiber can be further broken down into a structure called the myofibril. There are numerous myofibrils contained within any one skeletal muscle fiber. The myofibril is further broken down into a structure called the sarcomere, and there are multiple sarcomeres that are lined up all along the length of the myofibril. When we look at any one individual sarcomere, the sarcomere is composed of myofilaments, and the two myofilaments we'll consider today are the thick filament called myosin and the thin filament called actin. When we want a muscle to contract, nerves need to stimulate that muscle to contract. So the interaction of nerves and muscle gives us the neuromuscular system. The coordination and integration between the nervous system and the muscles of the body are what produces the muscle contractions that we see. In this image, the nervous system is broken down into two parts. First, we have the central nervous system. The central nervous system is composed of the brain and the spinal cord. The peripheral nervous system is composed of the nerves that extend from the spinal cord and make connections to the organs within the body. For our discussion today, we're going to consider the nerves that extend from the spinal cord and make connections to skeletal muscle to induce muscle contractions. The skeletal muscles of the body are composed of multinucleated cylindrical cells called fibers, which run parallel to each other. And if we take a look at this image of our biceps muscle, we can see the parallel arrangement of the fibers. We can also see the numerous motor nerves that innervate the skeletal muscle, the biceps muscle, to produce contractions. This fiber arrangement into cells is very different from the normal cell that we typically think of in the body, which has a cylindrical shape and a centralized nucleus. Again, this structure of the muscle fiber into this cylindrical shape with multi-nuclei all along its length is related to the structure-function relationship. The skeletal muscle fiber is wrapped by a membrane called the sarcolemma. This is a thin elastic membrane that encloses the fiber's cellular contents and represents the true fiber boundary. Within the fiber, there is the sarcoplasm. The sarcoplasm contains the nuclei, substrates and enzymes required for metabolism, and other specialized organelles. One of those specialized organelles is the sarcoplasmic reticulum, which is key for regulating muscle contraction. The sarcoplasmic reticulum is a highly specialized network of tubular channels that allows a wave of depolarization to spread rapidly across the sarcolemma and into the inner environment of the muscle fiber to initiate muscle contraction. The sarcoplasmic reticulum does this by acting as a storage unit for a large concentration of calcium within the fiber. In this image, we can see the sarcolemma, or the muscle fiber membrane, which wraps the entire muscle fiber. We can also see the sarcoplasmic reticulum. We can see that reflected in the blue mesh-like network that's wrapped around the myofibrils within this muscle fiber. And everything that is contained within the sarcolemma is within the sarcoplasm. In this image, we can see an enlarged view of the interface between a nerve and a skeletal muscle fiber. This interface is known as the neuromuscular junction. At this interface, electrical events, known as action potentials, are transmitted from the nerve to the sarcolemma on the muscle fiber membrane. The transfer of the action potential from the nerve to the skeletal muscle induces a series of events that ultimately result in muscle contraction. And this is known as excitation-contraction coupling, or EC coupling. In skeletal muscle, EC coupling describes the cascade of events initiated by an action potential starting at the motor nerve and ending at the surface membrane of the muscle fiber, resulting in the release of intracellular calcium from the storage within the sarcoplasmic reticulum and ultimately regulating whether muscle contraction takes place. In this figure, we see there are 9 individual steps that are needed to induce muscle contraction, and following contraction, induce muscle relaxation. In step 1, nerves stimulate muscles to contract through a chemical messenger known as acetylcholine. The chemical messenger acetylcholine ultimately is responsible for transferring the action potential from the nerve to the muscle fiber membrane. In step 2, we can see the action potential is fully transferred to the sarcolemma. In step 3, the action potential causes calcium to be released from its storage within the sarcoplasmic reticulum, and this is one of the key steps in regulating whether muscle contraction will ultimately take place. In step 4, in the presence of a large concentration of calcium, actin and myosin begin to interact within the sarcomere. In steps 5 and 6, force is produced through the continued interactions of actin and myosin. In steps 7, 8, and 9, we can see that muscle relaxation can be initiated following contraction. And this begins when calcium levels in the sarcoplasm begin to decrease, preventing further interactions between actin and myosin. Now we'll take a closer look at the actin and myosin interactions within the sarcomere. The sarcomere is known as the functional unit of the muscle fiber. And we can see we've talked about this breakdown already, where we have groups of muscle fibers representing our muscle, a single muscle fiber, and a line of sarcomeres that are lined up all along the length of that muscle fiber. It's the interactions of the myofilaments within the sarcomere that induce or are responsible for muscle contraction. We can see in this image, in this blue box, a single sarcomere represented. On the left side of the screen, vertically, we see one boundary of the sarcomere, and then on the right side, also vertically, we see the other boundary of the sarcomere. From these boundaries, the thin filament actin proteins extend inward toward the center of the sarcomere. At the center of the sarcomere, extending out toward the boundary, is our thick filament myosin. The sliding filament theory states that a muscle contracts because the proteins, or in this case the myofilaments we just talked about, within the sarcomere slide past each other. And we can see that depicted in this image. At the top, this sarcomere is at rest. We see the boundaries On either side of the sarcomere, the actin filaments extending inward and the myosin filaments at the center. When a muscle contracts, the boundaries of the sarcomere are pulled toward the center, and we can see at rest there is a lot of space in between the outermost boundary of the sarcomere and the outermost point of the myosin in the center, and this space is on both sides of the sarcomere. During muscle contraction, this space is progressively lost as the boundaries are pulled toward the center via the interactions of actin and myosin. At the bottom of the picture, we see a point where we have complete contraction of this sarcomere. The outermost boundaries of the sarcomere are almost in contact with the myosin at the center. Now, in this completely contracted state, this was brought about by, again, the interactions of actin and myosin, and we can see that reflected in the enlarged image on the right. Extensions from the thick filament myosin, called myosin heads, project up toward the thin filament actin. There are numerous myosin heads associated with any myosin filament within the sarcomere, and all of these myosin heads will interact with actin to produce the forceful contraction. Through an energy requiring process involving ATP, the myosin head will utilize the energy from ATP to physically move and pull actin toward the center of the sarcomere. On the left, we again see our biceps muscle. The biceps muscle is uniquely structured such that the fibers run the whole length of the skeletal muscle. The force of the muscle, in response to contraction of these fibers, is due to pulling on the bone at the points of attachment, and we have two attachments we can talk about. At the top of this image, we have the muscle origin. This is the location where the muscle joins a stable and immovable skeletal part. At the bottom of this image, we have the muscle insertion. This is the location where the muscle joins a movable skeletal part. For the biceps muscle, the origin is located in our shoulder, and the insertion is located in the elbow. So when the fibers contract in the biceps muscle, the insertion point at the elbow will pull on our forearm, allowing our forearm to move up toward our shoulder as the muscle contracts. Now we can visualize these muscle contractions in this video of an isolated piece of skeletal muscle that's being electrically stimulated to contract outside the body. In this example, electrical stimulation is being used to mimic the nervous system, and it's producing an electrical pulse once every second, and we can see the muscle responding to that pulse by contracting. The fibers in this muscle are running vertically the entire length of the muscle, and hopefully you can envision within those fibers the sarcomeres all lined up, shortening to produce the contraction that we see. We can also envision this by looking at this video and thinking about resistance training. In this example, this student is going to perform the bench press exercise. The bench press exercise utilizes the large muscles of the chest to move the bar from the chest to arms extended position. This movement is produced by the contraction of the muscle fibers within the chest. There are two main types of skeletal muscles in the body, and they differ in three specific ways. First, the way that energy is utilized to produce the forceful contraction. Two, the speed at which the muscle contraction occurs, and 3, the ability to resist fatigue with repeated contractions. When we look from person to person, there are differences in the structure and function of each type of skeletal muscle. The first muscle we'll consider is the fast twitch muscle. The fast twitch muscle is specifically organized to produce forceful contractions very quickly. The fast twitch muscle is composed of fast twitch muscle fibers. The fast twitch fiber's intrinsic speed of force development is approximately 3 to 5 times faster than the slow twitch fibers. Fast twitch fiber activation predominates in sprint activities and other forceful muscle actions that rely almost entirely on anaerobic energy metabolism, which means that these contractions are performed in the absence of oxygen. Activation of fast twitch fibers plays an important role in the stop and go or change of pace sports such as basketball, soccer, lacrosse, or field hockey. The second muscle we'll consider is the slow twitch muscle. The slow twitch muscle is composed of slow twitch fibers and is specifically organized to produce low force contractions that can be repeated over a long period of time, making slow twitch muscles ideally suited for prolonged aerobic exercise when there is no limit on oxygen delivery. Slow twitch muscles are very resistant to exercise-induced fatigue. And activation of slow twitch fibers plays an important role in aerobic and endurance events such as distance running, cycling, and swimming. Now, we can talk about these two muscle types in isolation, but it's important to realize that both slow and fast muscle fibers will contribute during near maximum aerobic and anaerobic exercise events. When we consider the muscle composition of untrained individuals, meaning people that aren't actively involved in exercise training or athletics, on average, there's 45 to 55% slow twitch fibers in the muscles of the arms and the legs. Larger muscle fibers in male athletes and a larger total muscle mass are the main gender differences in muscle. What this means is that male athletes will generally be able to lift heavier weights than female athletes because there's a larger muscle mass contributing to that lift. However, if we express the force production relative to the size of the muscle, that gender difference goes away and there's no difference in force production between men and women. While no gender differences exist in fiber type distribution, there is a large inter-individual variation in fiber type. Generally, the trend in one's muscle fiber type distribution remains consistent among the body's major muscle groups. However, certain patterns of muscle fiber distribution appear when comparing highly trained individuals. So, for example, a highly trained endurance athlete will possess predominantly slow twitch fibers in the majority of the muscles in the body. In contrast, highly trained strength or power athletes will possess predominantly fast twitch fibers in the majority of the muscles in their body. In this figure, we can see the percent contribution of slow twitch fibers to different sporting events and its association with oxygen consumption or the ability to deliver oxygen to those working muscles. At the very top, cross-country skiing and long-distance running are associated with high percentages of slow twitch fibers and a very efficient delivery of oxygen to those working muscles. In contrast, athletes that are involved in sprinting 100 and 200 meters or in weightlifting have a relatively lower percentage of slow twitch fibers in their skeletal muscles and a less efficient delivery of oxygen to those working muscles. In this table, we can see the relative involvement of muscle fiber types in a number of different sporting events. The type 1 fiber is what we referred to earlier as the slow twitch fiber. And as we can see in this table, slow twitch fibers are associated with performance in the marathon, in distance cycling, and cross-country skiing. In contrast, type 2 fibers are what we referred to earlier as our fast twitch fiber. Type 2 fibers are involved in sprinting events, weightlifting, football, baseball, and basketball. So if we consider these athletes that we started at the beginning of our lecture, it's important to understand that skeletal muscle is trainable. So these athletes didn't just become good at these events simply because of the genetic makeup of their skeletal muscles. There was training that was involved in enhancing the function of their skeletal muscles. So, with proper exercise training, we can become better at producing high force, powerful muscle contractions, or we can become very efficient at producing low force contractions that we can repeat over a long period of time. Now this occurs at the expense of, if we ask these two athletes to run a marathon, they will have a very difficult time completing that marathon, although they're extremely strong. And in a similar way, if we ask these two athletes to lift a heavy weight over their head, they would have a hard time lifting really heavy weights over their head, but they have the ability to run for long distances. So to answer our question, how does skeletal muscle contract to produce force? Nerves stimulate muscles via electrical impulses called action potentials. The action potentials are transferred to the skeletal muscles at the interface of the nerve and the muscle, known as the neuromuscular junction. Transfer of the action potential at the neuromuscular junction induces a series of events known as EC coupling. EC coupling leads to release of calcium and ultimately regulates the interactions between actin and myosin. Muscle fibers shorten or contract based on the sliding filament theory. So the myofilaments actin and myosin slide past each other within the sarcomeres, producing the contraction we see at the muscle level. The contracting fibers pull on the attachments to the bones. Muscle actions are associated with the type of muscle involved. Fast twitch muscles are used to produce high force and fast muscle actions, while slow twitch muscles are used to produce low force but repeatable muscle actions.